juncture. We've been working diligently with Austrianova to deal with the issues surrounding the growth of the cells from our master cell bank, both before and after encapsulation. And we've had some challenges that we had to overcome. We had a team of scientists in the United States working together. We actually added a new cellular biologist to our team from a very large biotechnology company in the US to solve or help solve or deal with the problems that, or the challenges that we were facing. We're here on the ground now in the beginning of the process, and we have our team here available to function as a resource to Austrianova on an as-needed basis. The team that we have here has been working with Austrianova for the last 6 weeks, solving problems that collaboratively were a better way to solve than individually. We set out to find a cellular biologist that had a particular expertise with the cells that we use in our therapy. And we wanted somebody independent of the company that manufactured the master cell bank, and we wanted somebody independent of Austrianova. We actually wanted somebody independent of our entire organization. We needed a fresh set of eyes to make sure that we hadn't missed anything. We have too much invested. We've spent a lot of time getting to where we are now, and we're at the ledge of jumping into a process that will enable us to complete our investigation on a new drug application and commence a clinical trial. There's no substitute for being here with Austrianova at the very manufacturing facility where all of the work leading up to where we are now has taken place. It's either been here or it's been in the Austrianova facility or laboratory in Singapore. But there's no substitute for personal contact and the relationship building that needs to go on, particularly with someone new to our team like David Judd, where he can see the facility, he can talk to the very individuals that were involved in developing this technology a long time ago with Bavarian Nordic. And it's a way for him to accelerate his learning curve that talking to somebody over a telephone or through a video conference can't achieve. We've seen that for the last few days. David and everyone here has made quantum leaps in their knowledge base in terms of what is actually occurring on the ground in Thailand by Austrianova. Absolutely. David is a brilliant cellular biologist, particularly with regard to growing cells, whether they're in culture or encapsulation. But more particularly, he has a specific expertise in the very type of cell that we use for our therapy. And so his input has been invaluable. He took our knowledge base to a completely different level and accordingly helped in the overall process to where we find ourselves today. We believe that given the tests that we've run on this entire process up to this point, that the cells will grow better, faster, and more efficiently than they were growing before we made the changes to our manufacturing process. In fact, we think the numbers will be better, the viability numbers will be better, and most importantly, the enzymatic activity within the cells will be better. We've seen preliminary test results from the experiments that were conducted in Singapore based upon the changes we made to both growing the cells and then post-encapsulation. And we're pretty enthusiastic about the results of the efforts that everybody's been making. If there was one critical change between the encapsulated cell product that was used in the earlier 2 clinical trials, it's the reproducibility of the capsules, the uniformity. And we think we now have an added value by having produced a master cell bank that grows better and has more enzymatic activity than its parental cell line from which the master cell bank was manufactured. Every time that you give a patient our encapsulated cells, they have to act the same way and do the same thing time and again without fail. And they have to be safe in the same way time and again without fail. We're doing 2 staggered runs to satisfy the person who will take responsibility for releasing the product into the United States. His level of confidence that it's a reproducible product on more than one occasion. So we decided to go ahead and do 2 staggered runs. We don't need the data from 2 staggered runs. What we need is the data from the testing of a run that produces positive results for us. Once we have that data, we will then incorporate the data into our investigational new drug application, which is the final piece that we need in order to complete the application. It's challenging in that the FDA requires a lot of information, a lot of data, and a lot of testing to go on before you're enabled to encapsulate these live human cells and put them in another human being. Safety being the most significant of the concerns. But of course for us, efficacy is as well. It not only has to be safe, it's got to work well. And working well means it has to have a very positive impact on the outcome of a patient with pancreatic cancer. And we think it will.